Hello everyone. So today let's begin with a new chapter, that is sexual reproduction in flowering plants. Okay. Now, when we are talking about sexual reproduction, we already, as discussed earlier, we know that it is a process of fusion of gametes, isn't it? It involves two type of individuals, a male and a female individual, or an individual which produces the two types of gametes, a male gamete and a female gamete. They will fuse together and produce a diploid zygote. Okay, so that particular process of fusion of gametes is known as sexual reproduction. Now, the sexual reproduction in flowering plants, that means the angiosperms. Now, when we are talking about angiosperms, that means these are the particular plants which are considered to be the most advanced type of plants among the plant kingdom, isn't it? That means we have studied in plant kingdom about the algae, bryophytes, pteridophytes, gymnosperm and angiosperms. So angiosperms, these are the most advanced type of plants in the plant kingdom. Now these angiosperms, they actually originated between the Cretaceous and Jurassic period of the earth and were mostly dominant during the Cenozoic era, for which that particular time period is considered to be the golden period of angiosperm. Now, when we are talking about sexuality, that means the reproduction process, sexually reproducing process, this was first of all studied by Jacob Camerarius. Okay, and all the process was properly discussed and explained in different steps. So before going in details to the reproduction process, let's first study about the structure of an angiospermic flower. Okay, now this is an angiospermic flower. Okay, you all are familiar with this diagram. Now these are the four holes of an angiospermic flower. This green color portion, this is the calyx. Okay, now see this green part, this is called the calyx. Okay, its individual unit is called sepal. The colorful part, this pink color portion which you can see, this is called the corolla and its individual part is petal. These are the androecium. Individual units are called stamen, and this is the gynoecium, that is individual units are called carpel, and this is the whole structure of an or else of a angiospermic flower. Okay, so these are the four holes of an angiospermic flower. Now the calyx, which is green in color, that means it will be definitely taking part in photosynthesis. This colorful part, it helps in pollination, attraction of the insect. This is the male reproductive structure, this is the female reproductive structure. So as we are discussing about sexual reproduction, so definitely we will discuss in details about these two structures of an angiospermic flower. Okay, now the male reproductive structure, that is the stamen, it consists of two parts, the anther and the filament. Okay, now this anther, it consists of two lobes. Okay, these are the two lobes of an anther. It is a knob-like bilobe structure, okay, tetragonal, and these two lobes consist of the pollen sac or the microsporangia. Now see, these are the pollen sacs. Okay, now here, as it is bisporangiate, that means each lobe or each chamber consists of two microsporangia. That's why a total pollen anther is tetrasporangiate. That means it consists of four pollen sacs in four corners. Now, if we consider or if we study in details about the microsporogenesis process, that means the process of formation of male spores, pollen grains, then first of all let us study about the structure, internal structure of the anther. Now, if we cut a transverse section of an young anther, then first of all we will get a mass of tissue, homogeneous cells, okay, a mass of tissue of homogeneous cells which is surrounded by epidermis. Now we know that all tissue system, it consists of an outer layer which is termed as epidermis. Now here in a young anther, the epidermis is surrounding a mass of homogeneous cells. That means these cells are similar. Now gradually when the anther develops, then what happens? 4 cells in 4 corners. Okay, that means now here the microsporangia, it actually develops hypodermally and 4 strips of archaeosporium develops. Now in this tissue, in 4 corners, 4 different type of cells starts developing. Okay, these cells contain a large nucleus and dense cytoplasm as compared to the other cells. Now these are all homogeneous cells, but just 4 cells are becoming clear in 4 corners. Now the main characteristics of these cells is that they have large nucleus and dense cytoplasm. We call these cells as the archaeosporial cells. Okay, this tissue layer is called the archaeosporium. Okay, the fertile tissue layer, and this particular cells are called the archaeosporial cell. Now gradually when the anther starts developing, these archaeosporial cells, they undergo transverse division. Okay, now in this diagram you can see the shape of the anther is gradually changing, and here the archaeosporial cells have undergone transverse division. The cells towards the outer side means the cells that are towards the epidermal layer, these are called the primary parietal cell. Okay, and those towards the inner side are called the primary sporogenous cell. So you can see that each 4 cell has divided into 2 cells, and in each cell, the cells towards the outer side are called the primary parietal cell, and those towards the inner side are the primary sporogenous cell. Now in between, a vascular strand develops. This is the initial of the vascular strand. Okay, the tissues become differentiated. This will later develop into the connective. Now in this diagram also you can see that this is the epidermis. The red line is indicating the epidermis. The 2 next lines are the parietal cells. That means the primary parietal cell is start undergoing division, and the sporogenous cells are also undergoing division. So you can see 3 cells here. Here also, that means 4 corners, 4 cells will consecutively start forming. Next, if we look in details, the next time what happened? The primary parietal cell, it undergoes repeated anticlinal and periclinal division. Now, what is anticlinal and what is periclinal division? When a division takes place, that means now suppose this is a particular cell. This is the base of the cell. If the division is taking parallel to the base of the cell, then this type of division is known as periclinal division. Periclinal division. Okay, that means the division takes place parallel to the base of the cell. And if periclinal division takes place, so definitely parallel cells are dividing. So what happened? The cells will grow in this form. That means it is responsible for the height of the plant. Okay. Again, if a cell divides perpendicular to the base, okay, this is the base, this is a perpendicular division, that this is known as anticlinal division. Got it? So if anticlinal division takes place, that means the cells will increase in this pattern. So here the girth or the width of the particular plant will increase. So due to periclinal division, the plant will grow lengthwise, and due to anticlinal division, the plant will grow breadthwise. Okay, that means its structure will, or its circumference will increase. Now in this structure, the primary parietal cell, it has undergone repeated anticlinal and periclinal division and has formed the 3-layered spore sac wall. Okay, this spore sac wall The outer wall is known as endothecium. Got it? Now, this word, theca, it represents the wall. Okay, so here we are using thecium. Endothecium means outer layer. The middle layer, that means the middle portion, and the inner layer, that is known as tapetum, which is the main nutritive tissue. Okay, so tapetum, this word is important because it is the tissue which provides nourishment to the developing microspores. Now, if the parietal cells are developing, that means what happens? Simultaneously, the sporogenous cell will also divide. So these sporogenous cells will undergo division and they will now behave as the microspore mother cells. Okay, these are all diploid cells. So these are all the microspore mother cells. Okay, now see, this is a proper structure of a mature anther. Okay, it consists of the outer epidermis. Okay, then four corners have four pollen sac. Each pollen sac consists of the sac walls. Okay, it consists of the endothecium, middle layer, and tapetum, and the inner cells are the sporogenous cells. Got it? Now this is the connective, that is the particular portion which vascular strand consists of the vascular strand. Okay, now this is the whole structure. This is one lobe, this is one lobe. Each lobe consists of 2 microsporangium. That's why it total 4 microsporangium is present, and this is known as the tetrasporangiate anther. Got it, the concept? Now the sporogenous cells, which after division forms a mass within the tapetum, they behave as the microspore mother cell. Now these microspore mother cells, they are basically actually twice and diploid cells. Now they will undergo meiosis and they will give rise to 4 haploid microspores or pollen grains. Okay, and during this period some spherical bodies are formed inside the tapetal cells before their disintegration. Okay, and these spherical bodies are known as the Ubich body, which are actually made up of sporopollenin. Now when we are getting the term sporopollenin, that means it is present in the exine of the pollen grain, and it is a particular layer or substance which is resistant to various chemicals and enzyme activity. That's why this helps in formation of microfossils. Okay, so the term sporopollenin is very, very important. Remember it. Now when the Ubich layer body is formed, gradually what happened, the tapetal layer starts disintegrating. That means like this type of thing happens. This is the sporangium, this is the microspore mother cell. This will divide and form a tetrad. This remains as a ball. Okay, it is enclosed within a callus layer. These particular balls are called the Ubich body. And when they are developing, that means they are now digesting or utilizing the tapetal cells for obtaining nourishment. Got it? Now, the Ubich layers gradually they participate in the formation of exine of the microspores within the pollen sac. Okay, now here only these cells will divide and form the pollen grains. So these pollen grains are now thick walled. Okay, these are also known as the microspores. Initially what happened, they remain together attached forming a tetrad with the help of a callus layer, but later what happened, that callus layer disintegrate or just dissolves, and finally by enzyme activity and it just releases the pollen grain. Then what happened, the whole sac consists of pollen grains. Okay, now this structure you can see here, the pollen grains, they are released actually. Okay, that means here when they are full, the inner tissues disintegrate and gradually they are released within the anther. Now when they are matured enough, then what happened, from this portion That you can see in this diagram, this portion, isn't it? This is known as the stomium layer. This yellow color portion, this is the stomium. Now here a slit occurs. This slit helps in dehiscence of the particular spores, microspores. Now when they are full, they will become turgid. Now when turgid cells will finally, what happen, they will burst out and they will release the pollen grains in these two portions. Then finally in the stomium a slit occurs, for which these are liberated. That means they are dehisced. Okay, now see here, this portion, it has just slit has occurred. Now this will gradually starts moving. That means the mature anther dries up and then a slit occurs, and with the loss of water the thick walls of the endothecium also breaks, and when it breaks it ruptures, it liberates the spores, and finally with the help of pollinating agents they will ultimately get dispersed from the anther to different portions. Now when pollination occurs, that means it will definitely have to fall on the stigma, isn't it? So either they will fall on the stigma of the same plant or to different plant. That means when this is matured, ultimately it breaks and liberates the pollen grains. So when the pollen grains are formed, that means the process of microsporogenesis is completed. Okay, so you go through this video, go through the diagrams and go through the notes also, study it properly. This process is very, very important. Microsporogenesis process, that means how the pollen grains are formed from one cell. It divides into two and then finally they repeatedly divide into the four pollen sac, which later matures and ruptures and liberates the spores. Okay, so this process of microsporogenesis, hope this video is clear to you, and if you have any problem then let me know.